is to suggest that the natural R&D department outside the door here has been innovating solutions to common problems that we also face for millions of years. And so it's really a treasure trove of opportunities or solutions to common problems that we might face. I just want to point out a few here. People are starting to use the hydrodynamics of whale fins to improve the efficiency of wind turbines. It really increases their performance by like 85%. To put these little tubercles on the leading edge of a wind turbine blade. Another good example of this is that the kingfisher was used as an inspiration for a real challenge that these bullet trains in Japan had, which were that they would enter and exit tunnels, and that transition from a tunnel to a tunnelless space acts a lot like the interface between water and air. They would create these sonic booms when they would go into or out of tunnels. Obviously not desirable for anybody living nearby or in the train itself, and so they changed the shape of the bullet train front to simulate how kingfishers do that with their bills, because they face a similar problem. New needles are being produced that are mimicking the biting mouthparts of mosquitoes, and this makes them less painful for injections. And of course, pharmaceuticals have been going to the natural world for natural products for a really, really long time. So biodiversity is really essential because it helps us to solve problems that are pressing for humans. And I'll get back to how jumping spiders connect into this in a little bit. As a biologist, kind of sticking my biologist hat on, when I think about biodiversity, I oftentimes gravitate towards certain groups that really are kind of showy, like these birds of paradise here, because I and other biologists have this intuition that these groups might have something to teach us about how this wild array of colors and songs and dances that these birds perform might arise. But in doing so, we oftentimes overlook other less showy animals, sometimes even in the same environment. So those birds of paradise cohabit with the frogmouths, and these are 3 species of frogmouths, and if you're not a super bird nerd, you're going to have a hard time telling them apart, right? So something really different is happening with frogmouths. They're not undergoing this kind of rapid radiation of color and song, et cetera. They're kind of staying pretty much the same, but they're in the same places. So why is it that one group goes bonkers and another group kind of quietly putters along? And when we think about this in evolutionary terms, we oftentimes do what we call tree thinking. And this is to look at relatedness in terms of a phylogeny, or a kind of a family tree of how different groups are related. And so a group that has rapidly produced a bunch of very different species in a phylogeny might look like this. Lots of species out here that have arisen very recently, so time is kind of going from the bottom to the top here. So this might be the birds of paradise here. And then other groups like frogmouths might just kind of be slowly adding to their numbers over time. Or maybe they're even dwindling. They might be like the horseshoe crabs of the world, where they had a heyday some point in geologic history and there's just a few left. And so these are the kinds of dynamics that we try to understand. And there's really 2 main approaches that we take to try to understand these. The first is to try to figure out, did something happen? Did something happen at this point here that really changed the trajectory of a group like those birds of paradise? So are there key events or major transitions? And the other is that we look at processes that might generate biodiversity within a group that might be the kind of grist for evolution. Now, I do work on birds sometimes, but typically not. And that's because these animals are arguably more fascinating, more beautiful, and easier to study. And actually, I've been excited about insects and spiders since before I can remember, since before I was even forming memories. And so today I want to tell you about work that my lab's been doing to study these jumping spiders. And we do this worldwide. It's taken us everywhere from Malaysia and Singapore to South Africa to India. We've got a planned trip in about 6 months to Brazil. And so we study these animals all over the place. Now, many people have encountered, how many of you have encountered a colorful jumping spider online? Online, seen pictures or videos of these spiders? Yeah, so they've really kind of come into the limelight recently. And that has led people to believe that jumping spiders are oftentimes really showy and colorful. But actually, most jumping spiders are pretty drab. They might engage in these kind of wild displays or unusual postures or dances like this jumping spider up in the top left here, which is a Phidippus jumping spider from North America. But they oftentimes lack that colorfulness that you see in some of the jumping spiders featured online. And so one of the questions that my lab is really interested in is why are some jumping spiders so brightly colored and others are not? Simple, basic question about these animals. And I'll point out that jumping spiders are incredibly numerous. There are more species of jumping spiders in the world than there are species of mammals, all right? So this is a huge group to be studying. It's as if I was saying, why are some species of mammals more colorful than other species of mammals? It's an enormous group worldwide. One clue to why these jumping spiders in particular are brightly colored is that you're looking at only boys right now. So these are all male jumping spiders, and males oftentimes, if they're brightly colored, use those bright colors in dynamic courtship displays to females. All right, this is part of their wooing or persuading of a female to mate with them. But this is a little bit more high stakes than what happens here at Urban Artifact on a Friday night. And the reason for that is because female jumping spiders are just as likely to eat him before he mates with her than they are to mate with him. You can see how tentative he is, right? So she's a voracious predator. It'd be like trying to persuade a tiger to be friends with you, right? Okay? And so part of the function of these bright colors is to persuade females not to eat him, to persuade females that he is the right mate for her, and to kind of buy him time. So we'll get back to this in a little bit. Now, one question I often get asked is why are jumping spiders suddenly kind of in the news? Why are we hearing about them just now? And the reason is that jumping spiders are really small. This is a real match here, not a stage prop. And this is one of those peacock jumping spiders that I just showed you a video of on the top of that match. So this is perhaps one of the reasons why Jen let me be on stage tonight, is because they're small and they're harmless. So they're typically the size of your pinky fingernail or smaller, and although they're venomous, they do have venom that they use to subdue their prey. They really couldn't get a fang into you if they tried. So they're really not threatening to humans, although they're great if you've got mosquitoes or other unwanted insects in your life and you want fewer of them. So here's what I want to go through today. I have kind of three parts. The first is to give you a primer on how jumping spiders see, because it's totally different from the way that we see. Then I want to tell you about some of our work on color vision and color signaling, and I'll end with how those dynamic displays that I showed you an example of engage with where females look in the world. All right, so let's start with how jumping spiders see. How many of you have interacted with the jumping spider in your life? Okay, wanted or didn't want to interact, but did nevertheless. If you did, the chances are that jumping spider that saw you might have turned to look at you. And this is a signature characteristic of jumping spiders. It gives them this sense of intelligence because they're very visually guided, and they really want to look back at you with the four eyes that face forward. So although they really do have the proverbial eyes in the back of their head, jumping spiders have 8 eyes. There's not an area around the hemisphere around them that they can't see you. They would prefer to look at you with these 4 forward-facing eyes. So that's why they swivel to look at you. And the reason for that is that these eyes, which will be the focus of this little primer tonight, provide them with the best vision, the best view of the world. So if I were to slice this jumping spider, I know it's not very nice to describe, but if I were to slice it through the top of the head, it would look something like this. Somebody else did the slicing. And so here are those 4 eyes. The ones with the big lenses here, those are called the anterior medial eyes for their position on the head. We also call them the principal eyes. And then these anterior lateral eyes with smaller lenses here, and those are also sometimes called the secondary eyes. So I might slip between those two languagings of that. So let's start with those secondary eyes, the anterior lateral eyes. Okay, what do they see about the world? This is a specialty of my lab at UC. And we use techniques that are oftentimes borrowed from or analogous to what your eye doctor might use to look at your retina. We use ophthalmoscopes. And one of the things that the ophthalmoscopes that we build for spiders, so we're kind of like spider eye doctors, one of the things about those ophthalmoscopes that allows us to do is to look in through that small lens and actually image the retina inside the head. So what you're looking at here, each of these, I don't know that you can see this from where you're sitting, but each of these is almost like a coffee bean shaped object here. Each of these little bright spots is what's called a photoreceptor. It's like the rods or cone cells in your retina that you're using to watch this presentation tonight. So we can see them in here, and if we move the ophthalmoscope or the spider around, we can begin seeing different parts of its retina, and we can stitch them together into a view of the full retina that gives us some information about how much of the world this retina sees and what kind of detail the retina sees. The detail part is also called acuity. And these lateral eyes, these secondary eyes, are pretty good, actually. You don't really have a mile marker for what 0.4 degrees is, but it's less than half a degree different in space will look like a different object. So if you had black and white stripes or a black and white checkerboard that was in 0.4 degrees in space, they'd be able to see that as a checkerboard instead of a gray. And I'll tether this to some other measures in a second. They have a big area of binocular overlap. That's this dark gray area here, and that may allow them to see depth in the world. I have a graduate student studying how these animals see depth. And another thing that we know about these eyes is that every single cell in them is green sensitive. So there's no color information coming in from these eyes. These are predominantly motion sensitive, modest resolution eyes that are giving them monochromatic information. So it's kind of like a black and white view of the world, okay? The big eyes in the front of the head, the ones that make these animals feel maybe a little cute, those are the principal eyes, and they're built completely differently. They're fascinating eyes. They actually have two lenses. And so the first is this lens on the outside. That's something that you see when you see a jumping spider. Behind that is a long fluid-filled eye tube that has what we call vitreous humor, much like the fluid in your eye. So light is focused down this long tube. At the bottom of that tube is a second diverging lens. So the first one's a converging lens, the second one's a diverging lens. That combination is what binoculars use or what a Galilean telescope might use, okay? And then at the very base of that is a retina, which I'll tell you a little bit more about. So what is the value of having these two lenses? Well, they work like a telephoto system. Actually magnify the image that this first lens produces. They magnify that onto the underlying retina. So they work a lot like binoculars do to kind of magnify the world in fine detail, okay? And that image is magnified onto these really strangely shaped, boomerang-shaped retinas that are actually tiered. So instead of having a flat sheet of photoreceptors like those anterior lateral eyes, these principal eye retinas have tiers, so light traverses 4 separate tiers as it moves through the retina. So here's light coming in through what we call that pit lens, and it's going to traverse 4 separate tiers of photoreceptor cells, okay? And you can see this boomerang shape here. Kind of drive this point home a little bit. Here's one of our ophthalmoscopes, and it's going to focus down in. You see the one of the first tiers come into view. That's going to go out of view, and we're going to focus deeper in the retina, and a second pattern of photoreceptors is going to emerge. That's a layer underneath it, okay? So this is what this looks like. We've got 4 tiers. Tiers 1 and 2 kind of extend all the way out into the boomerang wings, if you will. Tiers 3 and 4 are very central to it. And what's unusual about these bottom tiers is how tightly packed the photoreceptors are. They're actually at the optical limit of how small a photoreceptive device, whether it be in your camera or it be in a biological system, can be. And so the combination of those unusual lens optics and that tight packing of photoreceptors gives these eyes extraordinary resolution. So they can see objects as distinct in space if they're 0.07 degrees apart. Okay? It also means that they have really narrow fields of view like binoculars would or a telescope would, and we'll get back to that a little bit later. But let me put some of these numbers that I've just given you into context here. So this is a classic relationship in vision research. Here's body size on a log scale, All right, so we go everything from a fruit fly to a human. And here's spatial acuity, or the ability to see fine pattern in the world. So lower values of this mean better, sharper vision. Okay? So one of the kind of main take-homes here is that in order to get sharper vision, you need to be bigger, because you need to build bigger eyes, right? So small things have pretty poor vision. A fruit fly, crappy vision, would never be allowed to get behind the wheel of a car, right? 4.6 degrees apart in space is how far apart in space two things need to be distinct. That's about the width of a banana at, like, you know, finger's length, right? So they're pretty crappy at seeing the world. They do clever things to surmount that, okay? And you can feel really smug because you're at 0.007 degrees here in your fovea. This is why you can read things on a sheet of paper, all right? That's how you can probably read that from where you're sitting, hopefully. Okay? Don't be too smug, because there are things that are better than you, right? So falcons are better than you, and that gives them the sharp-sighted ability to see very small things like prey from a great distance, okay? So where do we place these jumping spider eyes? Well, 0.4 is already pretty good. So 0.4 places us over here. These are those secondary eyes, and they're as good as the biggest dragonflies get. And dragonflies are notoriously sharp-sighted insects with huge eyes. Jumping spiders typically would fit inside the head of this dragonfly, right? So they're already punching above their weight, as it were. But what about those principal eyes that I told you were really extraordinary? Well, they're right here. They're way off this common trend line here of needing to be bigger to see better. And they're not just extraordinary on their own right, but they're actually better than your house cat at seeing the world in fine pattern, okay? They're about as good as a chicken or a pigeon, and birds have really excellent vision, okay? So if you've got like a Chiweenie or something like that at home, it's seeing the world in worse detail than the jumping spider on the windowsill. And I just learned actually that elephants are about as good as a cat in spatial acuity. So there you go. You get a bunch of animals out there that you would think would see the world better than jumping spider, and they don't. All right, there's a couple of other things I want to tell you about here. The first is that this lens system here suffers from a problem, actually, that your eyes do as well, and that is that the lenses focus short wavelength light, ultraviolet, violet, and blue light, closer to the lenses than red light. This is something that your eyes do, and it creates a visual illusion where if you put like magenta and teal next to each other, it kind of vibrates. You have a hard time figuring out where things are. And so this is something that's really pronounced in these jumping spider eyes. It's actually magnified by that second lens, okay? And people have speculated that, well, maybe the way that they get over this problem is that they actually use that tiering that I talked about in the retina, where they would place the photoreceptors that were sensitive to long wavelength light, like reds, yellows, and greens, down here in these bottom tiers, and then the UV and blue sensitive photoreceptors maybe in the upper tiers. And in the kind of early 2010s, we got evidence that this was actually true, at least for one jumping spider. So that jumping spider from Southeast Asia had green photoreceptors in those bottom two tiers and UV sensitive photoreceptors in the upper two tiers. So this circumvents that issue of chromatic aberration. It also provides them with color vision. Okay, rudimentary color vision, but color vision nonetheless. So this seems to be an organizing principle of the retinal tiering, is that you should be putting the short wavelength sensitive cells closer to the lenses and the longer wavelength sensitive cells farther away. So you can already imagine this is a pretty complicated field of view that they're using to look back at you. They've got this motion sensitive monochromatic periphery, a lot like your peripheral vision, and then these foveal like things, right? A lot like your fovea, color vision, these weird boomerang shapes, okay? And that's the visual field that they're trying to inspect the world with. Okay, let's transition now to talking about color vision. I mentioned just a minute ago that they have color vision in those principal eyes, and we really began studying this in these animals. Habernatus pyrethrax, the word means fiery haired. It's for that bright red face. This is a handsome male here with his bright orange knees. We were just out in Arizona with the BBC. They're filming a special on these guys that'll be out, I think, 6 months to a year. Males are brightly colored, do all sorts of dancing. Females, also very cute, not so brightly colored. And they live in the Sonoran Desert. And I actually began working on jumping spiders with this species mostly as an excuse to go back to this habitat, because I did my graduate work in Arizona. This is a wonderful, vibrant ecosystem. And so what we do out in these habitats is we actually hunt for these little jumping spiders one at a time in these light-saturated environments like this Ocotillo forest outside of Tucson, and we're capturing them one at a time in little jars and bringing them back to the lab where we get to study their behavior. So we have about 400 of this species in the lab at UC right now. And one of the things that we're interested in is how males court females. So here's a courting male. He begins with these waving displays which alert her to his presence. He's showing off that bright red face. If he transitions in a little bit closer, then he's going to do some really sexy things like raising knees over the top of his back, doing these tarsal flicks up at the front. He's singing to her right now with vibratory songs which we can't hear. That air drumming right there, totally coordinated with the vibratory song that he's producing. It's going through the substrate that he's on, and she's hearing it with her legs. She has special organs in her legs called lyriform organs that allow her to hear that. But we're here to talk about color and vision. And so one of the things that a past postdoc in the lab did was to ask whether or not that bright red face that the male shows off early on in courtship, does that matter? What if we get rid of it? What happens? And so she got rid of it in some males. And this is trickier than you might imagine. Remember how small these things are. And they also are very, very delicate, sensitive creatures that if you try to use marker or paint or something like that, they swoon and die, right? So what do we have to use? Well, we had to use Sephora liquid eyeliner to paint off these faces. And let me tell you, that's a really weird conversation to get in with the Sephora folks at the mall. Like, what are you using this for? I'm painting my jumping spiders' faces. They tend to leave you alone. Little sidebar, when I was working really intensively on a species of butterfly that eats kale, I would buy kale from Whole Foods by the bushelful and never tell them what I was using it for, but I was always in there. And they apparently had some kind of office betting pool between was I making smoothies or enemas out of it? And I was like, what? No, obviously I'm feeding caterpillars with this stuff. That didn't really help. Okay, so Lisa blacked the faces of a bunch of jumping spiders and then compared their performance in courtship to jumping spiders that had not had their faces black, still had that red face intact. And the measure that you see on these graphs here is how close they could get to the female before the female either attacked them or got bored and walked away. And it turns out that males with red faces intact were able to get closer to females. So they were able to progress through that courtship sequence farther than males that had had that removed. Females lost interest in them much more quickly or attacked them at higher rates. But this was only true in full sun conditions, so under bright light conditions. And I'm going to bookmark that for you because we'll come back to it later. In the shade, you can still kind of see that pattern if you squint, but it wasn't statistically significant. All right, so maybe color is important to these animals in courtship, right? Male red may be important to his courtship success. So we moved from there to thinking about what these animals eat and whether or not they're using color to make decisions in that context. So these are voracious predators. They're what are called cursorial predators, which means that they hunt on the run. They don't build capture webs. They can make silk, but they just make these little silk sleeping bags for nighttime purposes. But then they abandon them and they go hunt during the day, and they're really clever. Actually, the Chinese word for jumping spider translates to fly tiger, and they do route detouring. They can count. They do all these really cool things that are probably selected for by their predatory lifestyle. So we wanted to know, maybe whether or not they were using color, because lots of things in their environments are colorful. Has anybody ever seen one of these harlequin leafhoppers on a hosta plant? You should look for them. They're like broadleaf plants. They're native here, brightly colored. Some of the things that are brightly colored in their environment, like a milkweed bug, are really toxic. Actually, if a jumping spider attacks one of these things, they'll just drop it because it's so toxic, because they sequester compounds from the milkweed. So what's that?
I said, aren't they kind of big?
They are kind of big when they get to be adults, but when they're little, they're tasty little morsels. So we did an experiment which we, in the lab, called the Taste the Rainbow experiment, where we colored crickets. These little pinhead crickets, anybody a reptile enthusiast here? All right, so pinheads, the tiniest little stage of cricket. If you've got like chameleons, you need pinheads. If you've got lizards, you probably don't. Turns out when they're this small, you can just put food coloring in their water, and after a couple of days, they're so translucent and they take up enough of that food coloring that they get to be colorful. So this is a really clever way that Lisa came up with to try to investigate whether or not there were color-based preferences amongst food items in these jumping spiders. And so here's what we found. Jumping spiders, the Habernatus pyrethrax jumping spiders, attack the red and yellow crickets at lower rates than blue, green, or control crickets. Control crickets were just kind of given a mix, so they still looked brown. Again, this pattern was only true under full sun conditions. So the challenge with this, for somebody studying vision, is that these different colors could look like different brightnesses too. So there wasn't a way to disentangle brightness from color. And that's important for scientists studying this stuff, to really say it's color and not brightness that these animals are using. So we switched to a different paradigm where we tried to train these jumping spiders to learn to either avoid or prefer red as a color in their prey. All right? And we did this by using those milkweed bug nymphs. You can see some of them up here, teeny tiny. And the nice thing about milkweed bugs is that you can raise them on milkweed seeds and they sequester all that toxic stuff and they're gross. Or you can raise them on sunflower seeds, no access to the toxic compounds, they're perfectly palatable. And so we raised jumping spiders on one of these two diets, and they all had access to these white-eyed fruit flies. And then one group just got the beige diet, which is just all white-eyed fruit flies. So nobody went hungry, but everybody was learning something different or not learning at all anything about red. And so following up on that, we said, all right, we've got our crickets. We're going to ask you if you prefer to attack the brown cricket over the red cricket, right? And so for red aversion, they should attack red crickets less than brown crickets. This is what we found. For the ones where the red prey item had been fed sunflower seeds and was perfectly palatable and actually easier to catch than those little fruit flies, we found that they had adopted a preference for red. And so they were attacking red crickets at higher rates. And then the ones on the beige diet were depressed and didn't care. No, they attacked it in both directions. So they'd never had an experience with red before, so they didn't have any any learned response to this. So this really cemented our understanding that these animals are using color and that they actually can learn to associate profitability or toxicity based on the color of their prey items. So we think this is really exciting. We've got a bunch of projects in the lab focused on this because there's lots and lots of little tiny, very colorful things in the world. And the dominant paradigm has been to think of these things as being shaped by birds. Birds have been like the dominant insect predator that people have thought about for a really long time. And birds think and see and act really differently from a jumping spider. So we actually think jumping spiders are shaping some of these teeny tiny things that are way too small to be of value to a bird. And some kind of circumstantial evidence for that is that a whole huge group of insects from all different kinds of insect groups across the world make a living by pretending to be jumping spiders. So they actually spoof jumping spiders by evolving patterns that look like jumping spiders. And some of these patterns actually completely fool the jumping spiders and scare them away. All right? So it's clear that these animals and these communities are responding to jumping spiders as a major predator. Okay. So color's important in foraging. Color's important in courtship. But I mentioned earlier that the evidence that we had about how they saw color was that they had ultraviolet sensitive and green sensitive cells in those principal eye retinas. And the problem with that understanding is that that would take a picture like this and make it look like this. A visual system based on ultraviolet and green sensitive cells can't see red or yellow or orange as being a distinct color. To kind of tether this to a real life example for some of you, this is the reason why you can hunt in blaze orange. Is because deer have blue and green sensitive cells in their eyes and can't see blaze orange as being distinct from the background, and so their world looks a bit more like this, right? So we went from collecting these animals in the beautiful Southwest to dim basement room in the outskirts of Baltimore where we worked to learn a technique called microspectrophotometry from the guy who's done all of the work on vision in mantis shrimp. I don't know if anybody has heard of a mantis shrimp, but if you have, this is the guy. His name's Tom Cronin. And so we learned this technique from him. And the way that this works is that you section the retina, you slice the retina up. Each of these little dark spots here is one of those photoreceptor cells. And then you measure how those photoreceptors absorb light. And that tells you something about what light they would be sensitive to and therefore would respond to. And that can give you some understanding of their color vision. So we spent a couple of weeks kind of listening to our tans whisper away in this dark room. You have to do it in the dark because you don't want to bleach the retinas with light. And what we found was they have ultraviolet photoreceptors in those upper two tiers and green receptors in those bottom two tiers. And we hadn't gotten anywhere because that's what we already knew. It hadn't solved the puzzle. We were really like, and this was 2 weeks of like 12-hour days in under dim red light. You know, we're like getting creative about Spotify playlists, but we're really kind of losing our minds. And the guy that we're working with, I call him up, and he's this wonderful but very curmudgeonly gentleman. And he's, what is it? You know, I was like, well, Tom, you know, like we're not discovering anything here. We're just finding the stuff that we already knew, and it doesn't solve this problem. Wow, let me come down and look. So we've got one of these sections up, and we've sectioned it and handled it completely under red light to avoid bleaching any of the cells in the retina. And Tom just flicked on the white light, which we would have never done because it would have ruined a sample. And what he saw was this little ruby red spot. And it turns out that what Tom stumbled upon for us was really the key to understanding how these jumping spiders see color. So this is actually a red filter, a bit like red sunglasses, built into the retina. And it's in the path of light. Remember that light has to go through all 4 of these tiers, and it's between the bottommost tier and the second tier here. And what it does is it prevents, just in that tiny little spot, it prevents anything but red light from getting to the bottom tier. So although those bottom tier cells are normally green sensitive, there's no green light for them to respond to. All they can do is respond to red light. And so that shifts their functional sensitivity off into the red. So it's a kind of a clever way of creating a third category of photoreceptive cell that gives them red sensitivity, but it will only work under high light conditions. So those results from our courtship experiment and our predation experiment, where we only got those responses to color under bright light and not in the shade, that's because they're using sunglasses in their retina basically, and they can't see red when the lights go down. Okay, so right now here, a jumping spider would not be able to see these lights as being red here. So that takes this picture back to here and opens up this whole realm of color for these jumping spiders. Okay, so we wondered if this was a major transition. Who else in the jumping spider world has this red filter? Nobody knew it was there. They were looking, maybe they're looking past it. Maybe all jumping spiders can see this. So we went and started looking for this red filter. And what we found was that it was restricted to jumping spiders in the genus Habernatus and very closely related jumping spiders. We sampled a bunch of other jumping spiders that were more distantly related, and they didn't have a red filter. Okay? And so this we began to really understand as being this key transition, this evolutionary event that opened up this whole world of color to this group of animals, this evolution of this red filter, opening up this world of color to these animals. And the subsequent response has been, now there's this whole world of color that can be used to show off, to create differences, to make choices about what to eat. It provides them with all sorts of fodder for, so I'm just showing you, these are different species in that Habernatus genus here. And I'll get back to Habernatus americanus and Habernatus halleni a little bit later because their courtship is really cool. So let's go back to this though. Nobody here, does anybody here know what the genus Moratas is? Probably not, right? So this is a totally different group of jumping spiders. It's in blue here just so you notice it. And that's because they don't have a filter, but they are the peacock jumping spiders that I started off with. brightly colored, no filter in their retinas. What the heck's going on? So we did that microspectrophotometry technique again, and what we found was that instead of using a red filter, they had evolved two new classes of photoreceptors, yellow and blue sensitive ones. So they're tetrachromatic. They have 4 separate color channels. We only have 3, so they're doing better than us. They're about as good as birds. All birds are tetrachromatic. So UV, blue, green, and yellow sensitive photoreceptors. Okay? So that kind of takes this diagram and starts making us think, there's multiple major transitions. And so at this point, we began going out looking. Where's their color that might hint that there is something going on with color vision? This is another one that we went to India for this one. This is called Luserti's rainbow spider. And this is a male. And you can tell that they're using these colors to communicate with each other because they show them off to each other, but they're also forward facing. So predators from up above are not able to see them because they're pretty drab. And this might be one of the reasons why you might not have noticed some of these Habernatus spiders being so colorful, because they do live in your backyards around here. So Stenolurus luserti, that's the scientific name for this guy. Again, independently evolved. They're totally separate group. They'd be like the difference between giraffes and elephants in the mammal tree. That's how far apart this is from those peacock spiders. Has independently evolved its way to color vision, slightly different peak sensitivities, a blue and a red sensitive photoreceptor type. And so we're adding these up as we study these around the world. Jumping spiders seem to have repeatedly evolved improvements to their color vision with filters, with changes to the types of photoreceptor cells that they have. And the one thing I want to point out, these are little diagrams of how those are arranged in the different tiers, is that they're all organized by that guiding principle of that linear chromatic aberration. So they're putting all of their short wavelength sensitive things close to the lenses, long wavelength sensitive things to the kind of very bottom of the retina. And there's lots to discover. This is a genus Mexigonus. We don't know anything about it. They live in Mexico. We're just describing the species. So, there's dozens and dozens in this group. We don't know how they see the world, so this is what we're chasing around the world. So we're really beginning to understand why some jumping spiders are so colorful and others are not. And at the basis for this, at least in part, is these changes to their ability to perceive color in their world. And this is all stuff that we've discovered in the past 3 or 4 years. So the world did not know that jumping spiders had anything but UV green dichromacy until just a few years ago when we started working on it. So super exciting stuff. All right, in the last couple of minutes of my talk, I want to talk a little bit about these dynamic displays and how these animals look around the world. And I think you can think about this a bit like how you kind of navigate a world that's really complicated. Here's like a recreation of an old Times Square, and as you look around this complicated scene, your eye is probably drawn to different things, to flashing lights, or maybe to the face below arrow collars. You can understand this scene a bit based on what we see. So red's in here. This is not a scene designed by your dog, right? Your dog can't see red at all. And so we know quite a bit actually about how humans see complicated scenes. Decades and decades of eye tracking work has really helped us to understand the guiding principles of human visual control. And one of the things that we've learned is that some of these things, the ways that we look at complicated scenes, are under cognitive, kind of active cognitive control, and some of them are not. We sometimes just have routines about how we explore something, and so one of the interesting things about this, though, is that outside of humans and dogs and primates, we know almost nothing about how animals look around the world, even though animals do a lot of looking around. Okay? So, of course, for jumping spiders, Times Square looks a little different. This is a complicated scene for a jumping spider, and so we're interested in how these displays in particular are responded to by the gaze of the female. How is she looking at this as it's happening? And you might be kind of scratching your head like, what do you mean look around at this world? And that's because I've concealed for dramatic effect one last feature of those big eyes in the center of the head, the principal eyes. So if you remember, those little red wedges there are the view of the world that they have. It's tiny. They are like binoculars. They're only seeing a very small part of the world. And that's a real handicap, you know. They'd have to be doing this all the time, and they don't. They just sit there and look at the world. And the reason for that is that they're actually able to move the retinas around inside the head to look at different points in space. Those blue bands that you see here, those are muscles that give them an almost 60-degree range of motion inside the head. They're moving the whole eye tube around inside the head. Now, I know you're imagining this with me right now, but I'm going to show you what this actually looks like, because it's crazy, okay? So here is a transparent juvenile spiderling here, and those things moving around inside its head are its retinas at the end of its those long eye tubes. It's tracking that brush. There's a little fruit fly for scale, so this is a really tiny little animal here, okay? I don't know that you can see it very well here, but there's a little dot moving around inside its head, and people oftentimes wonder what that is. That's something it ate. So if you can imagine moving around like sausage-sized things inside your head, everything else moves around. Their stomach moves around, their brain is moving around, okay? It's a good thing they've got an open circulatory system, or things could get really bad. Okay, so now when these animals mature, they melanize their head capsule so that it gets dark, and we can't look in from outside. And we're interested in adult responses to these. So we go back to some of those ophthalmoscopic techniques in collaboration with Beth Jacob and her lab at the University of Massachusetts Amherst. And so we can look in through the lenses with infrared to image the retinas, while at the same time playing them videos on little video screens. It's like a Clockwork Orange thing, but without the violent scenery, right? You know, so we tether the animals. They look down through a tube at something like this scary dot moving across the screen, and then we can actually track where those retinas. You can see those two boomerangs. And one of the things I hope you notice is that they can go right and left and up and down, but they also have these twisting motions that we don't really fully understand, but we think might help them to see things like lines in the world, or may even help them with depth perception. So if they look at something really exciting like a cricket, they do a lot more of those torsional, twisty movements there. And so we're working to try to understand what females are looking at when males are doing this. Now, we can't do this with the female live out there moving around. She's tethered. She's got to watch, I don't know. It's not pornography. It's foreplay, right? But videos of handsome males doing things like this. And so here's a video where we've tracked where the center of those retinas are. Remember, that's where their color vision is, right in the center, and that's where their highest resolution is. So she looks around it. And you know what's really funny is that I know this is anthropomorphizing, but when these females get bored, they've got like a 10,000-yard stare. And for this female, it's different for each female based on how they're aligned. It's often the kind of bottom right. So she's bored now. She's not looking at anything. And he's going to raise a knee, and she's going to see it in those lateral eyes that doesn't have to send a message back to the brain that something's moving, that send it to the muscles that cause her to look over. So let's see if we can run that again now that you know what you're looking for. Hang on a second. Oh, come on. Well, here's what this looks like. So this is the start points here are when that knee goes up, and you can see with some delay, because there's like, you know, the message needs to go back to control the muscles to send them over there. Then there's this flurry of activity that occurs looking at those knees, okay? And so we're able to do lots with this. We're able to look at these dynamics over time. One of the ways we can do this is to flatten over time and just look over space and see where females concentrate their visual attention when the male is displaying to them. And that's what this little heat map is here. You can see a lot of attention gets paid to those knees up there. You know, another thing that's nice about this is that we can do like, you know, a kind of deadpan version of this with the male and all of the right colors, or we can, you know, really go crazy and put the colors in different places, or we can remove some of the motion, et cetera. So we can manipulate these, because this is being played back on a screen, and that lets us tease out what is it about this male display that really seems to capture and captivate females. Okay, So here's what some of these results look like. These are going to be ones flattened over time. So this is just where in space do females look. And when my postdoc, Daniel Zurich, sent this to me for the first time, he didn't have the picture of the jumping spider underneath it. And it just totally blew my mind, because even without that visual aid, I already knew where the male was. I knew what his display looked like. I knew where every part was. Those little wrist flicks that he does, you can see that there's concentrations of female attention there. I knew this was where the knees were going to be raised. So what I'm saying is that by just knowing where females look when the male displays, I can see the fingerprint of the male display on it. Fascinating, all right? We can remove those knee movements. Females spend a lot less time looking up here. Intriguingly, females spend a lot of time kind of anticipating where those knees might be, so they spend a lot of time looking just at the top of the head where those knees might appear. So we actually think that some aspects of female cognition may have been shaped by evolving with these male displays. How females go about, this is this kind of like unconscious way of processing that caused you to lose the chicken game there. Females may have the same kind of stereotyped way of looking at a male display, and that stereotyped way may have been shaped by millennia of interacting with males with displays like this. We can get rid of color, and then suddenly females begin paying attention to really high contrast things. And I want to show you, this is where I need to transition. I want to show you a different way of looking at this, which is what we call a space-time cube. And let's see, I need to reload this. Oh, but it's not going to project over there. Let me see if I can get it over there. And I'll have to, yay. A little bit difficult to control. So this is a really fun thing to play with, because we've labeled different events here. And so you see the wrist flicks bring a lot of female attention up to that part of the male display, up on those raised legs. But then the male raises his left knee, and all of the females, each of these hot spots here are where the majority of females are looking. He raises his left knee, they go look at that. He raises his right knee, they go look at that. And then some of them go back to looking at the wrist flicks. Some of them stay down here still looking at those knees. And it turns out that color in this situation is responsible for how long females look at things. So if you remove color, females lose interest in those areas of the display. And so that seems to be really critical. The color is really critical for keeping female attention. So we're spinning this out, not just in Habernatus pyrethrics, but across this really species-rich group here, because these animals inhabit really different environments and they have very different displays. And so we're doing this kind of gaze tracking work, looking at different display types, and I'll show a little bit of a montage at the very end about what these displays look like, to better understand how these displays have evolved to capture, retain, manipulate female attention to advantage males. And so I'll just kind of end on a montage of what some of the variety looks like in this group. Even just in this one group of jumping spiders, this is Habernatus Hersudus, named for the hairiness of his legs here. He's an arboreal species. We think that twitchiness of his display there helps to set him off in terms of motion dynamics against the kind of swaying of the mesquite bosques that he lives in. This guy here is Habernatus halleni. We call him Mr. Sparkle Pants in the lab because he's got these iridescent legs, and he does a lot of this, like, throwing up his legs to flash at females. And he's going to do a kind of like sidling display that's very reminiscent to me of some of those birds of paradise, where he kind of flattens his body and shows this really iridescent pattern here. He's going to come back across. You can do it, man. Oh, he's going to come back into view. There you go. Ta-da. Yeah, so he's got great moves. Some of these animals are just really kind of creepy. If you could imagine running into this guy at the bar, he's just got this like lurker phenotype. This is Habernatus oregonensis. We call him Popeye, for the obvious reason. But this is what he does. This is his deal. He just kind of lurks there, looking swole. And then some of them are like really just twitchy. This is not like a playback issue with my computer. This is really Habernatus americanus here, named for his red, white, and blue. And this is, yeah, he's just a spaz, going hard on the red side of things. So these are a real delight to have in the lab and to study in the field. We're actually really intensively studying some of these species out at the edge of Appalachia Preserve. Really wonderful sites out there in the cedar barren habitats that are out there. Really unique habitat type that with high levels of endemism. So if you haven't been out there, you should definitely do it. Okay, a couple of closing thoughts. So hopefully I've persuaded you that we've identified some major transitions in color vision that help us to understand some of the biodiversity in this group. And that processes like visual attention on the part of females can really shape the solutions that males arrive at in trying to persuade females that they are the mate for them. So I mentioned really early in the talk that we should think about the benefits of studying biodiversity from the standpoint of what they can do for us. And I think that's a fine way of thinking about things. Jumping spiders are not going to help us with bullet trains, but I'll tell you about some of the things that, that they are helping us with. One is thinking about how to create incredibly small, incredibly high performance cameras, which have a huge market in the cell phone industry, right? So the new iPhones are really working off of some of the same principles as jumping spiders with that telephoto lens and then the wide angle lens working together to provide a seamless view of the world. That moving retina is actually directly inspired some of the imaging technology that's up on Mars right now, one of the Mars rovers. It gives increased acuity to that. Jumping spiders are not only tiny and not threatening, but they have really small brains. And they have 8 eyes and massive amounts of information coming into those. Their brains are the size of a poppy seed. And they have to make these really fast decisions on behavioral timelines about what to do, where to jump to, et cetera. And so we're working with Uber and some other self-driving car groups to try to understand this is a problem that self-driving cars have. The one that you're looking at right here does pretty good on the road. It's got a liquid-cooled supercomputer in the trunk. It's worth like $60,000 to $75,000, right? So you get rear-ended in one of these things, and that's a huge expense. So how do we downsample and do clever algorithmic things to try to extract really important visual information from a bunch of sensors? This is something that perhaps jumping spiders can teach us a bit about. And of course, biomedical research really benefits from having high performance imaging technologies that you can swallow or that can be put into a vein or used in robotic surgeries, et cetera. And so again, we're thinking about miniaturization. So this is a reason why you should care about biodiversity. And I would be telling you a tall tale if I told you that this is the reason why I do what I do. Because it's not true. Like, I care about this, it's important, but the real reason that I do what I do is because I'm curious. And I just want to say that curiosity is an essential part of what it means to be a human. We are curious animals. It's something we should embrace. It's something that many of us leave behind when we're this age. We say, that's for kids, and now I do serious stuff. But I think we do that at great peril. And I actually think, oftentimes curiosity-driven science is a pejorative. It's like, that's not really that serious. It's got no real application. I actually think curiosity-driven anything is really what it is about to be human. And so the reason why I do this stuff is that I'm curious. I still feel like that little kid inside. It's why I'm doing all of this work, is because I'm wanting to discover new things about the natural world. So just a shout out to curiosity being something that we should really foster in children and in ourselves as adults. So with that, I haven't done this in a vacuum. There's tons of people that have been involved in this. I'm just here to tell you about it tonight. So folks from around the country and around the world that have really enabled this work. The National Science Foundation has supported a large share of this, and really appreciative to them and to you as taxpayers for helping me and my lab and collaborators to discover this stuff. And the University of Cincinnati is a wonderful place to do this kind of work. And we have people at high school, undergraduate, graduate, and even retiree levels that come and work in my lab. So if you're curious about doing this and you would want to spend some time doing that in the summer times or any time really, feel free to reach out to me. And I'm happy to take questions at this point. But yeah, that's what I have to share with you tonight. Yeah.
I have 3 questions. If that's
Okay. Yeah, absolutely. I'm going to repeat them because there are people maybe watching online at some point.
My first question is, do other animals have like the lower receptors, the lower frequency receptors near the cornea or on the outside, I guess? Is that common amongst all eyeballs?
Okay, so the question is, do other animals have kind of lower acuity or lower frequency photoreceptors in the periphery? Is that kind of what you're talking about?
The magenta and then the green.
Oh, the tearing.
Yeah, the tearing.
Yeah. So other spiders do, but and then there are animals with compound eyes like butterflies that have tiered what are called rhabdomes. But there, the tiering isn't really accounting for that. It's more like how do you get color information out of a facet that looks at a particular point in the world. So there is retinal tiering out there, but not in like large vertebrates, for example.
So humans don't.
So humans do not. Nope.
And my second question is there any other animal that does like the filtering, like where there's like the rose-colored lens?
Yes, the question is whether other animals do the same kind of filtering, and the answer is yes, absolutely. So all birds have what are called oil droplets. Those don't create novel classes of photoreceptors, they just refine what wavelengths a particular kind of photoreceptor is sensitive to, and that actually allows them to see a greater variety of colors. So it kind of separates the bands of color that each photoreceptor is able to see. Turtles have them, lots of reptiles do. Again, butterflies also have filters, but they're a bit more like the spiders in that they take a green sensitive photoreceptor type and they filter it with red, and that shifts its sensitivity to longer wavelengths. So insects are using this, and birds, for example.
My third question is kind of philosophical, I guess. Is like, how do you determine? You mentioned like the females have like this behavioral thing that happens, and you attribute it to cognition. How do you determine if something is cognition versus just automatic response?
Okay, so the question is, how do you, like for an example in spiders, determine if something's cognition versus automatic responses? It's a good question. The view used to be that, that especially insects and spiders were just little automatons, and they just had like knee-jerk responses to things. And so there are lots of ways of querying that. One way of doing that is to set up experiments where animals have to choose between 2 options. And so therefore, they need to evaluate one option and another option and then select the one that's most beneficial to them. And those can actually get incredibly complicated. And so oftentimes that's done in jumping spiders, for example, by giving them 2 different routes. And they have to assess the route and figure out which route is going to get them to a place faster or more reliably or without risk. So you can put all sorts of kind of variables there. It's kind of like mazes, but for jumping spiders, right? And so that suggests a level of strategy and cognition. And actually, jumping spiders will select routes that take them out of view of their eventual goal. So they can say, I'm going to go there, and then they can go without being able to see where they're headed for stretches of time and still follow the route. So that suggests a level of kind of planning and memory that we generally talk about in terms of cognition. In the context of courtship, for example, a lot of the gaze tracking stuff is suggesting to us that females are looking at different parts of the display before they make a decision, and they're making a decision about which male they might mate or not mate with. That's a loaded decision for them because they'll make that decision maybe 1,000 times over the course of their life, but they may only pick 1 or 2 males to mate with. And so which male are they going to do that with? It's a multivariate problem. They can't just simply say, bright red, that's my guy, because that's going to apply to 99% of males in the population. So when it comes down to females tending to prefer very subtle things about males that have the higher frequency knee raises, and those knees are orange, and they've got the right spot here, and we've got evidence that the females are looking around to see all of that, almost like they're checking boxes, that feels a bit more like cognition, where they've got a series of kind of thresholds or preference functions that they're using to apply to decide whether or not a male is acceptable. So I think it's a matter, I think we could probably argue about where that line should be drawn in terms of complexity. but we're oftentimes assessing very complicated decision making, which frankly some humans can't do. So so I think that the hegemony of like humans being the ones with cognition and everybody else, that's pretty clearly not true. But when we get into the point where, well, jumping spiders can plan a route that takes them out into the future and out of sight of where they're trying to go, well, that's pretty sophisticated. Some primates have a hard time doing that. So the lines get really blurry and we begin to talk about it as cognition. Does that help? Yeah. Yeah.
I have 3 questions too.
Okay, great. I'm here all night.
The first one is really stupid, but you said that their eyes work like Galilean telescopes. Are they inverted? Is the image inverted?
Yes, absolutely. Yeah, so the question is, do the eyes work like Galilean telescopes? And if so, are they is the image inverted? And the answer is absolutely
so
yes.
Processing everything upside down in the
Well, don't we see that way too? I mean, it's projectile.
Yeah, absolutely. And so there's a part of their what we call visual neuropills, the part of the brain that begins to process it, that inverts it again for them. Yeah, like us.
Second one.
Yeah.
Does the environment they live in, the average light level and the environment they live in, make a difference? Like you said, you're looking at ones that were from the Sonora Desert. I've been there. Very different from Ohio.
Yes.
So that ability to see better in bright light, would that be more advantageous to those spiders than ones living in my backyard?
Okay, so the question is whether or not the light environment that the jumping spiders live in influences what they see, how they see, what they do with their vision. And the answer is absolutely yes. It's also not one that I have the full answer to. It's something that we're intensively studying, because when we go worldwide, when I mentioned Singapore, we're in like virgin old forests, old world rainforest in Singapore. That's a very low light level situation compared to the desert. One thing that we've noticed is that filter-based mechanism of improving color vision only seems to evolve for species that live in high light environments. It's evolved twice. So there's a group in South Africa in the genus Hylis and a nearby genus, totally independent groups that seem to have evolved it, but they are bright light animals. And so some ways of improving your color vision may be restricted to circumstances where there's loads and loads of light. All right, so that's one thing that I can point to right away. But there are probably subtler things. So the color of light in forests is not equal. If you live in forest gaps that are high light versus in deep forest understory, and there are jumping spider species that are specialized for both things in any forest around the world, or if you're an underbark specialist like Platycryptis undatus, which we have around here, the light that you experience over the course of your life is going to be very different, and that's going to really shape the properties of your eyes. So one thing we think is that maybe many jumping spiders see color poorly because they tend to be in lower light habitats. It also seems to be what they inherited from the ancient jumping spider lineage, so they would have to evolve something different. But low light habitats seem to not select for improved color vision, simply because it comes at a cost to sensitivity.
Okay, my last one is, what's
Yep.
the difference in visual acuity between the males and females?
So the question is, what is the difference in acuity between males and females? So we've studied this only in one species, carefully. It's one that's really common here. It's called the audacious jumping spider, Phidippus audax. If you're at a dog park and you see a little black and white jumping spider eating flies on the fence post, is probably Phidippus audax. Females have better acuity than males do as adults in Phidippus audax. But they keep pace with each other through almost the entirety of development. The females do 2 more stages of development beyond the males, though. So they get to be bigger because they molt 2 more times. And the result is that as full-grown adults, females are bigger in body size, and their eyes are bigger, and their acuity is improved as a result of it. So yeah, other than that, so size is going to have some influence on the acuity of these animals. And there are size differences. Oftentimes females are bigger than males, and so that probably edges them out against males a little bit. But otherwise, we don't see a whole lot of evidence that males have like extraordinary visual acuity compared to females, or vice versa. It's pretty subtle. Yeah.
I had 7 questions, but they asked that.
Excellent. All right.
You mentioned that they can count.
Yeah.
But I don't know if maybe I missed it, but can you elaborate
Yeah, so colloquially we say that some species of jumping spider can count up to 4. So this is an assessment of their decision making to see whether or not they pick something that's not simply just more versus less, right? So if they get a decision to go one way or the other in terms of prey items, they seem to be able to detect the difference between 1 and 2, and 2 and 3, and 3 and 4. And if you kind of switch the order of those things in a way that would lead them to like just pick more versus less, they still seem to kind of track the numbers. After 4, it's just lots. I'm headed that way to eat. So this has all been done in a very clever jumping spider called Portia fimbriata. It's actually a spider hunting jumping spider. And so they're incredibly clever because their prey is really dangerous. So they have to make very careful decisions. And if anybody's ever read a science fiction book called Children of Time, if you haven't, it's a really cool one, but it's all based on Portia fimbriata. And it's this like kind of science fiction imagination into what a really hyper intelligent jumping spider society would look like. So, yeah. Yeah.
Lifespan and number of broods that they might have?
Yeah, so lots of jumping spiders will live to be about a year old. Some of them will live to be a couple of years old in the wild. In captivity, they can live between, 1 and 3 years, depending on the species. Jumping spider broods are really variable. So Phidippus audax is a great local jumping spider. Females there, they'll live sometimes 2 years in the wild, but oftentimes just a single season. They'll lay 3 to 5 egg sacs, which they don't tend to. They just lay them and then let them hatch, of 60 to 100 spiderlings in each. Okay, so that's a lot. It could be like 500 or 600 offspring that they produce over the course of their lifetime. Whereas some of these Habernatus jumping spiders, there are some that live in kind of sand dunes along the west coast, and they lay big fat singlet eggs one at a time and bury them in the sand. And so that jumping spider might only lay 15 or 20 eggs over the entirety of its lifespan. So they're really different. And the ones that are like way at the low end of the spectrum in terms of the number of eggs they produce and the number of offspring they produce are these ant mimicking jumping spiders. They're called myrmecomorphs. And we have some around here, a couple of species around here. Look carefully at ants, and sometimes it's a spider acting like an ant. But they're really narrow-waisted because they're trying to look like ants, and their abdomens are really tiny, and they just don't have the room to make very many eggs. So they're another group that just lays a couple of eggs at a time. And so we call this fecundity as scientists, and their fecundity is very low in comparison to something like Phidippus audax. So, yeah.
The basic lineage for spiders, do you have any idea when that arose?
Yeah, so jumping spiders are a pretty recent group in the grand scheme of things. So we're talking, like 500 million years ago. So major expansion of jumping spiders after the last glacial event. The Habernatus jumping spiders are probably about 90 million years old. So very recent to have produced 120 or so jumping spider species. So yeah, last glacial maxima was when jumping spiders as a group really proliferated worldwide.
Are you talking the Wisconsin glaciation?
What's that?
The Wisconsin glaciation?
Yeah, sure. Yeah, absolutely. Well, in every continent around the world, as those glaciers receded, then suddenly we get this kind of rapid invasion of new jumping spider types into all these different habitats. Yeah. Yeah. So they're a really cool lineage. They show up in amber, Baltic amber, and things like that. So.
Now, recently the filter rose. Speculation.
Yeah, that's a good question. Probably 100 million years ago, but we don't know. We don't have a good date for it in the second one. The first one is actually a fairly large group. There's this tribe, the Harmochirinae, that includes Habernatus, Pollines, and a handful of other groups. So that's a larger group, but that has the red filter. But then this Hillis group that I mentioned in South Africa, it's Hillis and Nigerella, just a couple of genera, and we don't really know. We're still working out. So a lot of this work's being done in collaboration with the world's expert and evolutionary relationships and phylogenetics for jumping spiders. He's at the University of British Columbia in Vancouver. And we're partnered with him because we're collecting all this material, and it's helping us to be able to begin dating these events and really solidifying these relationships. So we're kind of working hand in hand to try to figure out the time scales on these things.
So it's probably were independent events.
Yeah, absolutely. Yep. They're certainly independent events because of the number of taxa, the number of groups of jumping spiders in between that don't have the filter. It's kind of remarkable, actually, because you would think that it might be a complicated thing to turn on some kind of pigment production in your retina, but maybe not. This suggests that the lightning can strike twice on this front. Yeah. Yeah. All right. Any other questions? Yeah.
Do we know anything about how they're able to comprehend between the primary and secondary eyes? Like seeing having a telescopic lens and then basically just a very peripheral.
Ooh. So the question is, how do they kind of integrate the information from the different types of eyes? That's $1 million question. It's more than $1 million question. We have no idea, actually. We're just beginning to get into that. We know parts of the brain where the input from those eyes comes together. One area of the brain is called the arcuate body, and it's very similar to something that we know quite a bit about in insects called the mushroom body, which is an integration center for multiple senses. The mushroom body is where olfactory information and visual information and some other forms of information come in and are integrated in memory formation and navigation and decision making in insects. And the arcuate body is similar in some ways to that in spiders, although the connections are really kind of shaped differently. So people have gone in there with electrodes, and they can find cells in the arcuate body that respond to stuff that is seen in both eyes. So we know that that information is integrated in that portion of the brain. But how that's accomplished is not very well understood. And it's going to be some time because some of it's what we call neuroanatomy. It's like looking at how things are connected to each other in those areas. Some of it is electrophysiology, so measuring responses in those areas. The electrophysiology is hard in these animals because their body's under a positive pressure because it assists in their locomotion. They actually kind of hydraulically pump themselves around the world. And so it's a bit like trying to measure something with a needle inside a water balloon. Like as soon as you pierce that, then they kind of bleed out. And so it requires some really tricky stuff to try to, it's just trickier than in some other animals where things are not that kind of situation. But we're making strides there.
You mentioned the mushroom body?
Yeah.
Is that because it resembles mycelium or some other reason?
So the mushroom body is called the mushroom body because it's mushroom shaped, like your classic, toadstool shape. Yeah, so it has nothing to do with mushrooms really, except for some neuroanatomist was like, that looks like a mushroom, and there it is, yeah. So, and I don't know why it's called the arcuate body. If anybody knows what arcuate means, you know more than I do. All right, yeah.
So I was just trying to catch something. So they're trying to figure out what's going on inside of a brain.
Yep.
That's the size of a poppy seed.
Yep.
Inside of a creature that's like a lot
Yeah.
and wants to explode.
So the question or comment is that you're trying to study something that's the brain the size of a poppy seed inside like a little head capsule that's tiny and wants to explode. It's like very tricky. Well, it doesn't very well. One way of doing it is to not worry about things bleeding out and instead to immerse the whole head in like a little kind of aquarium, basically, where you just kind of put the head in something that mimics their hemolymph, their blood, so that it doesn't matter if they bleed out because it's submerged. So that's one way of doing things. The other way to do things is to try to cut a small hole that's so small that the surface tension of their hemolymph prevents them from bleeding out. But the problem with that is that then you're in a situation where you can't see what the hell you're doing. And so trying to poke something precisely somewhere and know where it was and what you were measuring is really difficult. And that's really what people have been doing, is creating these tiny holes, sticking an electrode that's not a sharp electrode, so it's not going inside a particular cell. Instead, they're using approaches, kind of signal-to-noise approaches or machine learning approaches to do what's called spike sorting. So they just get this like buzz of activity in that region because the electrode is just measuring the electrical activity in the whole area, and they try to look at patterns in the responses to figure out what individual units are doing out of that kind of, you know, TV noise, if you will. So, yeah. Yeah.
How often did the females completely destroy or get in the way of your males' experiments?
So how often do the females completely destroy or get in the way of our experiments? Well, about 20% of the time to 25% of the time, he's a snack. We break that up. We don't let that go all the way to belly. Yeah, no, we've got brushes and we shoo them off. But yeah, somewhere to 25 or 30% of the time, a female will go and try to attack a male rather than let him mate with her. And that's actually influenced by hunger state and whether or not she has mated before, right? So if she's already mated, she's got all the sperm she needs for the rest of her life, then every male's a snack, right? So, yeah, so female state, and actually males, at least in other spiders like wolf spiders, males can actually tell if a female's hungry based on chemicals on her silk that she leaves behind as she moves around the world. And that doesn't mean that males aren't foolish and don't try to court her anyway, but at least they go into the situation knowing that she's a little hangry and like things might not go as planned. So we don't know if that's true in jumping spiders, just simply because the experiments haven't been done, but I wouldn't be surprised if males are pretty savvy to the state of the female. But yeah, I mean, I know it's a trope, but it's true that like these jumping spider males, they'll court anything. They court dead females in the lab. They court females of other species. They're just like, I don't know, maybe today's my lucky day, you know? So yeah, but anyway, females are aggressive pretty frequently. So it's part of the equation. Yeah.
And mate just once, did you say?
Once, sometimes twice. Yep. Yeah, which is different. So I studied butterflies quite a bit too, and butterflies will mate 6, 7, 8 times sometimes over the course of their life. So that allows you the opportunity to study the patterns of what they prefer or don't prefer. It's a little trickier when it's like only once ever. So that's actually rare. In the animal kingdom, you know, most females will mate many times over the course of their life or have many partners. And I would say monogamy in the way that we conceive of it is rare. But jumping spiders are one group where females are largely genetically monogamous over the course of their lives. So, yeah. All right. Well, I'm here for a little bit longer. As long as the sky holds, I'm here on a motorcycle, so I need to scoot back to Clifton in case it gets rainy. But I'm happy to answer more questions as a sidebar. So, all right. Thank you.
Thank you. Before you head on, I'll tell you about next month's event. But first, a shout out, as always, to Urban Artifact, and a special thank you to Andrew and Devin of Radio Artifact, who are responsible for producing tonight's event. We are now streaming and recording our events. You can find those at YouTube, Radio Artifact, and at Twitch, Radio Artifact. In addition to this event and others in the future, they're producing lots of great independent music that you can find there, so do check that out. Next month, July the 6th, we are happy to have Bob Genheimer from the Cincinnati Museum Center. Bob is the curator of anthropology, and his topic is going to be the policeman and the privy. And this is some historical anthropology Bob was involved in doing some digs in downtown Cincinnati and uncovered quite a few interesting artifacts, including a full policeman's uniform and other pieces of belonging to that individual. And it's quite a true crime story. It is very intriguing, and there will be violence included. So please join us Wednesday, July the 6th, 6 o'clock, back here at Urban Artifact for that. You can always find us at Facebook at Science on Tap Cincinnati and at Meetup at Community of Reason. And one final comment, Nate made the comment about curiosity. And that is certainly a key value of the Community of Reason. Curiosity is a value in and of itself. And we all need more of that. So thank you again, Nate. Thank you all for coming tonight, and we hope to see you next month.